Welcome to the AP Bio Show, the show that's about your success in AP Biology. On tonight's show, we're going to get deeply into Unit 1, the molecules of life, with a focus on carbohydrates and lipids. We're also going to look at some biology in the news. We'll look at the Hayflick limit, which is the thing that keeps us from being immortal, that keeps cells from only being able to divide a certain number of times. And we'll have some of our regular segments, a weekly quiz, your questions, and tips for learning the greatest of all subjects, biology. About the AP Bio Show, we're going to be here on YouTube Wednesdays 6:30 to 7:30 Pacific Standard Time. The segments of the show will include amazing biology, biology in the news, a lesson of the week, a song of the week from Science Music Videos. There'll be a biology quiz. There'll be time to answer your questions, and we'll talk about how you can crush it in your biology course with learn-biology.com. A little bit about me, your host. My name is Glenn Wolkenfeld, also known as Mr. W to my students, and this year you're my students. That's because I'm a retired AP biology teacher, recently retired about 2 years ago. When I taught in the classroom, I had a huge amount of success with my students, most of whom got 4s and 5s on the AP Bio exam, and I'm looking forward to, through this show and through the other resources that I'll share with you, generating the same level of success for you. I'm the creator of the Science Music Videos YouTube channel, where you can find all kinds of lectures and songs that'll help you learn biology. I'm also the creator of learn-biology.com and the Biomania AP Bio app. Let's start with the lesson of the week, carbohydrates and lipids. Macromolecules are the large molecules that make up living things, and as an AP biology student, there are 4 macromolecule families that you need to know about. Those are the carbohydrates, the lipids, the proteins, and the nucleic acids. In today's lesson, we're going to take on carbohydrates and lipids. Next week, we'll look at proteins and nucleic acids. So let's start learning about carbohydrates. Carbohydrates include foods like sugars and starches, and also plant fiber like cotton. The monomers of carbohydrates, their building blocks, are monosaccharides, and the monosaccharide that you'll learn most about is glucose. That's because glucose is the fuel of life. It's the input for cellular respiration, and it's the product of photosynthesis. Glucose's chemical formula is C6H12O6. Here's its full structural formula. An abbreviated structural formula, you have to imagine the carbon and hydrogen atoms at each angle vertex, and a chain form. In solution, which means in cells, in life, glucose is almost always in a ring. Fructose is another monosaccharide that you should know. It's a structural isomer of glucose, which means it has the same empirical formula, but a different structure. Fructose is naturally found in fruit, where it attracts fruit-eating animals, which eat the fruit, swallow the seeds, and then defecate those seeds far away from the parent plant. That's an important adaptation for plant dispersal. We humans love fructose and other sugars. That's because our distant ancestors were tree-living primates who ate fruit and passed that preference on to us. Today, most of our fructose comes from corn, and up to 10% of the calories in a North American's diet can come from this monosaccharide. Monosaccharides are the monomers of carbohydrates. In my video about monomers and polymers, I compared monomers to Legos that can be put together to create larger structures. Let's see what happens when we start combining monosaccharides. When enzymes link 2 glucose monomers together through a dehydration synthesis, we get maltose, a disaccharide. Let's focus on the bond between the 2 glucose monomers. To do that, we're going to use a system that biochemists have devised where they assign a number to each of the carbons in a monosaccharide. Here you can see the numbers for each carbon in glucose. When enzymes link the 2 glucose monomers together to form maltose, they connect the number 1 carbon of one glucose with the number 4 carbon of the second glucose. That's called an alpha-1,4 glycosidic linkage. And the prefix glyco is widely connected with the carbohydrates, and we'll see it again throughout this unit. What do you need to know about that bond? Well, the main thing is that it's a covalent bond, and it has a specific shape. And we have enzymes that can break that bond, changing maltose into 2 glucose monomers that we can absorb into our cells for energy. Here's the disaccharide sucrose. It's table sugar, and it's what we put into our coffee and tea. Sucrose consists of a glucose connected to a fructose, and the bond between them is an alpha-1,2 glycosidic linkage. What do you need to know about that? It's another covalent bond. It has a different shape. We have enzymes that can break it apart, releasing glucose and fructose that our cells can absorb and use for energy. Sucrose, in other words, is a digestible sugar. The last disaccharide that we'll meet is lactose. It's the sugar in milk, and it consists of a glucose linked to another monosaccharide, galactose. All mammals drink milk as infants. That's what makes us mammals. In order to digest the lactose in milk, we need to be able to break this beta-1,4 glycosidic linkage between the glucose and the galactose, and all mammals are born with an enzyme that enables them to do that. It's called lactase, and it takes lactose and breaks it apart into glucose and galactose. Now, a lot of adult humans, including me, are lactose intolerant. That means that as adults, we don't synthesize the lactase enzyme, and therefore we can't digest lactose, and ingestion of lactose leads to all kinds of indigestion, cramping, flatulence, diarrhea, nasty stuff. Lactose intolerance might seem like some unfortunate disease, but when you think about it, lactose intolerance is the norm for mammals. You only need to digest lactose when you're a suckling infant. Those adult humans who can digest lactose are the inheritors of a genetic mutation that arose thousands of years ago in a certain population, and that mutation keeps the lactase enzyme active throughout adulthood. In those populations where that mutation arose and spread, a whole wide array of dairy food sources was opened up, and that became quite a beneficial adaptation. For those of us who haven't inherited that mutation, don't despair. There's an entire industry producing lactose-free dairy products and also lactase supplements so that you can eat lactose and digest it without a problem. If you want to learn more about lactose intolerance and the evolution of lactose tolerance, there's more about that at my website. Now let's turn our attention to carbohydrate polymers, which are known as polysaccharides. We'll start with starch. Starch is a polymer of glucose, and it's used by plants for long-term energy storage. That's why you find starch in plant parts like potatoes, and also in seeds like beans and corn. The simplest form of starch is amylose. It's just a straight chain of glucose monomers held together by an alpha-1,4 glycosidic linkage, the same one that we saw when we were discussing maltose. Another form of starch is amylopectin. In addition to chains, there are also branches between the number 1 carbon of one glucose and the number 6 carbon of another glucose. That bond is called a beta-1,6 glycosidic linkage. To digest amylose or amylopectin, we have enzymes that can break that alpha-1,4 or beta-1,6 bond, releasing the glucose monomers. That's why starchy foods like potatoes are loaded with food energy, and that's why potatoes were the key to astronaut Mark Watney's survival in the science fiction novel and movie The Martian. Here on Earth, starchy foods from plants like wheat, rice, beans, and corn, along with potatoes, are the staple foods that power human civilization. Glycogen is an energy storage molecule that's made by animals that's structurally very similar to amylopectin. In fact, it's sometimes called animal starch. That subscript indicates the number of glucose monomers that are chained together in that part of the molecule. Glycogen gets stored in huge molecular complexes like this in our liver tissue and our muscles. When our blood sugar level gets low, our bodies will convert stored glycogen into glucose. On the other hand, after a carbohydrate-rich meal, our bodies will take excess blood glucose and convert it into glycogen. This interconversion is controlled by hormones like insulin and glucagon, and breakdown of blood sugar homeostasis can cause diseases like diabetes. There's a lot more information about this on my website. Cellulose is another polysaccharide that's built from glucose monomers. It's the primary component of plant cell walls, and because of that, cellulose is the most common biological polymer on Earth. Cellulose is incredibly useful. Cotton fiber, wood, paper, they're all composed to varying degrees of cellulose. Like starch, cellulose is a polymer of glucose, but the glucose monomers are held together by a beta-1,4 linkage. Unlike starch, We and many other animals don't have enzymes that can break that bond. That explains why a cup of potatoes, mostly starch, has about 10 times the food energy as a cup of celery, which is mostly cellulose. We just don't have the enzymes that can break these glucose monomers off the cellulose chain and feed them into cellular respiration. You know that cellulose is strong. That's because the cellulose strands form hydrogen bonds with one another, and the collective effect of those hydrogen bonds creates a strength that we see in things like rope and wooden boards. Mammals like this deer have evolved mutualistic relationships with bacteria that have enzymes that can break the glucose monomers off of cellulose, making its chemical energy available. Other mammals like goats and sheep and cows have these same mutualistic bacteria, and they live in a digestive chamber that's called a rumen. Collectively, these animals are called ruminants. Among the insects, termites have evolved a mutualistic relationship with a protozoan that enables them to digest the cellulose in wood. That's good for the termites, but it's disastrous for the billions of dollars of damage to structures in the United States alone each year. So what are the key takeaways? Carbohydrates consist of monosaccharides. Those are the monomers. They're used for energy sources and building blocks. You see glucose on the far left. They also consist of disaccharides, also sugars. Those are used primarily for energy transfer. They include molecules like sucrose, cane sugar, and lactose, which is milk sugar. And there are polysaccharides. Those are used for energy storage, such as starch and glycogen, and they're used for structures. Cellulose, which is the key component of cell walls and which makes up plant fiber. Now let's move on to lipids. Lipids are characterized by the fact that they're wholly or partly nonpolar. They're not composed of repeating monomers. Think about what we just talked about with cellulose and starch. Glucose after glucose after glucose. You don't see that repeating structure in the lipids. There are important building blocks, but not monomers in exactly the same way. Their functions include energy storage and insulation. We see that in fats and oils. Waterproofing, which we see in waxes, membrane formation in phospholipids, and signaling, which happens with the steroids. One of the key characteristics of lipids is that they're hydrophobic. They're hydrophobic because they're mostly or entirely nonpolar. You know that oil and water don't mix, and that's represented here in this photograph. Here you can see a diagram that's showing water molecules forming hydrogen bonds with one another, but they're not bonding with the lipid droplet that they're surrounding. You can see the nonpolar nature in this structural formula over here of a molecule of fat. And you can see that it's almost entirely carbon, hydrogen, hydrogen, methyl groups essentially all around it. There is a carbonyl group over here, but the molecule is predominantly nonpolar and hence hydrophobic and hence won't bond with water. Fats and oils are the first subcategory of lipids that we're going to look at, and they are loaded with chemical energy. You can see in this food label over here that 1 tablespoon of oil has 120 calories. A tablespoon of butter has a little bit less, 100, and that's because of the water content of butter. When animals need to store energy, they put on a lot of fat, as you can see in this bear, which is about to go into hibernation. Fats are also used for insulation, and that's why marine mammals like the whales have a lot of fat on their bodies. It's hard to believe this, but whales were almost hunted to extinction to extract the fat from their bodies, which was used to light lanterns throughout the United States in the 1800s. Good thing that we don't do that anymore. Chemically, fats and oils are both classified as triglycerides. The glyceride part of the name relates to the fact that part of their structure is a 3-carbon alcohol that's called glycerol. That glycerol is bound to 3 fatty acids. Fatty acids are an important structural component of many lipids, and they consist of a hydrocarbon chain, that just means carbon, hydrogen, and a carboxyl group, carboxylic acid functional group at the end. Combine those two together and you get a triglyceride. That's a dehydration synthesis, and you have to remove 3 water molecules in order for that to happen. Fats are saturated oils are unsaturated. And what that means is that in the hydrocarbon chains in a fat, there are no double bonds. Every carbon is bound to as many hydrogen atoms as it can, whereas there's at least one double bond in the fatty acids that make up an oil. That makes them unsaturated. Because of that, fats are solid at room temperature, whereas oils are liquid. And what's important to emphasize is the room temperature part of that. You know that if you throw butter onto a frying pan, it'll melt. But fat molecules, because they have straight change, which results from the fact that the fatty acids are unsaturated, can actually line up close to one another, and very weak bonds can form between these adjacent fat molecules, and that's enough to solidify fats when they're at room temperature. By contrast, oils are liquid at room temperature because the double bonds bend the hydrocarbon chains, preventing oil molecules from getting close enough to one another to form intermolecular bonds. The second class of lipids that we'll discuss are the phospholipids. Phospholipids have a hydrophobic, non-polar tail. That's this part of the fatty acids that make up the molecule. Like triglycerides, there's also a glycerol in the middle, but there is a hydrophilic or polar head. And you can see a phosphate functional group that's over here. Phospholipids are often represented by some kind of diagram like this, and that is because of the role that they play in membranes. When you mix phospholipids in water, they spontaneously form hydrogen bonds with the water on one end. That's what the head does, whereas the tails will bond together to form a water-free zone. So here you can see this, water on the outside, water on the inside, and a water-free hydrophobic zone over here. That can lead to the formation of structures like this, little bubbles of phospholipids, and that is essentially the structure of the cell membrane. It's thought that when life was first forming 3.8 or so billion years ago, the spontaneous formation of structures like this that could encapsulate other biological molecules, nucleic acids, and proteins was an essential step in the origin of life. We'll talk much more about phospholipid bilayer formation when we talk about cell membranes in Unit 2. It's fabulous stuff, and we'll talk about the origin life in Unit 7. Here's a very cool application. Variations in the phospholipid structure can serve an adaptive function in browsing mammals that forage in snowy environments. What's that all about? Here we have an elk, and it's in the snow over here. Now, it's not uncommon among mammals, which maintain a constant body core temperature, for the extremities to get colder than the core. You experience that all the time whenever your hands or feet get cold. There's an adaptation related to phospholipids that's really extraordinary. The phospholipids that are in the hoof, that are furthest away from the core of the body, tend to have more unsaturated fatty acids, whereas those that are closer to the body core have more saturated fatty acids. Why is that? Because it's the same thing we talked about with the liquidity of oils as opposed to the solidity of fats. Because these phospholipids over here are more unsaturated, that makes the membranes more fluid, and that allows for the diffusion of materials through the membrane. It's a great adaptation, and it occurs at a molecular level in these mammals that are browsing in the snow. Now let's move on to steroids. One important function of steroids is their activity as hormones. Hormones are molecules that are released in one part of the body, diffused with the bloodstream, and then affect cells in another part of the body. And here you see a steroid hormone entering into a cell, binding with a receptor, passing into the nucleus in a complex with that receptor, and then activating genes. You see the DNA over here. That DNA becomes transcribed into RNA, and that RNA gets translated to become protein. This activation of genes leads to long-term and permanent changes within the structure of the organism, something that you recently experienced, because during sexual differentiation, it's the hormones estrogen over here and testosterone that play either a feminizing role or a masculinizing role. And that happens in two phases during life. It happens early in embryonic development, and then it happens again during puberty. Notice that it's only the difference between these functional groups over here, a hydroxyl group over here and a carbonyl group over here, that determines whether the organism becomes masculinized or feminized. Biology, always so amazing. Another steroid to know about is cholesterol. Cholesterol structure is shown over here. Cholesterol forms an important part of cell membranes. You can see one integrated over here, 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 and so on. What cholesterol does is it stabilizes membranes and it acts as a kind of fluidity buffer. What that means is that when it gets too cold, what cholesterol does is it keeps the membrane fluid, and when it gets too warm, it keeps the membrane stable. It keeps those phospholipids from flying apart from one another. That's the role of cholesterol. The last lipid that we're going to discuss are waxes. Here's the structure of a wax. A long hydrocarbon chain connected to another long hydrocarbon chain. Waxes are incredibly abundant in the biosphere. Every single leaf, every blade of grass that's in every ecosystem is covered with a waxy cuticle that's shown over here at number 1 and down below. The wax is creating a waterproof layer on the top and the bottom of leaves that keeps water from escaping. And as you know, waxes are also used by animals. Here's the honeycomb of a bee, largely composed of wax. Wax is super important. Key takeaways related to lipids. Lipids are completely or mostly nonpolar and hydrophobic. One category of lipids are the triglycerides, that includes fats and oils, and they're used for energy storage and insulation. Phospholipids are key membrane components. You'll remember that one side is hydrophobic, the other side is hydrophilic, so they form these phospholipid bilayers. Steroids can be chemical signals, and as cholesterol, they're used in membrane stabilization, and waxes are used for waterproofing. You just had a lot of information come at you related to carbohydrates and lipids. To really internalize and deeply understand this material, you've got to interact. And to interact, you should go to learn-biology.com and try the tutorials. You won't regret it, and you won't believe how much you'll learn. One of my goals for the AP Bio Show is for it to be a live show, but stuff is happening in August and I can't be here live. But I promise that if you leave questions and comments, I will answer everyone in a timely manner. So please leave questions, please leave comments, and I promise I'll answer. Now it's time for biology in the news. We're going to talk about the Hayflick limit, cell senescence, and telomeres. The reason why we're talking about this is we're honoring Leonard Hayflick, who lived from May 20th, 1928 and died on August 1st, 2024. Hayflick discovered why we in our cells don't live forever. He identified a phenomenon that's actually come to be known as the Hayflick limit. Let's see what that story is all about. Cells are the basic unit of life. Here's a very simplified one that's shown over here, and cells form tissues. Tissues are groups of cells that perform a similar function, and then a variety of tissues organized together to form organs. Here we see the skin. These are the levels of organization. You can learn more about that through a tutorial on learn-biology.com. Tissues can also be artificially grown in a culture dish. Here you see cultured cells growing in a petri dish. Now, a long-held belief was that when cells were grown in culture, they were essentially immortal. Hayflick, through a series of experiments in the 60s and 70s, proved this to be incorrect. Eukaryotic cells, they reproduce through a process called mitosis. And during mitosis, one parent cell becomes two daughter cells. And during this process, the genome of the cell, the genes are stored in these units that are called chromosomes. Those chromosomes are doubled and then they're pulled apart so that all the genetic information in the parent cell is passed to the daughter cells. And again, that's passed through the transmission of chromosomes, which are long strings of DNA. What Hayflick discovered was that for most cells, for example, the cells that make up your skin, the cells that make up your internal organs, the number of times that a cell could divide is limited to about 50 cell divisions. And after that many divisions, the cell became senescent. That's probably a new word. And a senescent cell has no more cell division, and ultimately it dies. That prefix, SEN, if you think about the word senile, it's the same prefix. So it's loss of function. Why does that happen? The first thing to note is that the way that our DNA is organized, we're eukaryotes, that's our clade, a concept we talked about last week, but that's a clade of all the organisms that have nucleated cells. Our chromosomes are linear. In other words, they have a beginning and they have an end. This form over here shows replicated chromosomes, but just imagine one beginning, one end over here. And DNA, as we'll learn later, it's a sequence of units that are called nucleotides that spell out the proteins that our cells are supposed to make. In our prokaryotic cousins, the DNA is circular. In other words, it's a loop. There's no beginning, there's no end, it's a circle. Linear DNA can't be completely copied. We can't really go into the details of this now. We'll deal with this when we get to Unit 6, when we learn about the details of DNA structure and DNA replication. But just know for now that during the replication of linear DNA, the kind that's found in eukaryotes, a little bit of the end gets lost in every replication cycle. In other words, the DNA gets shorter and shorter. That's called the end replication problem, and it's unique to eukaryotes. Eukaryotes have a partial solution to this problem. The end of a chromosome has a bunch of repetitive DNA. It's non-coding DNA. It doesn't code for proteins, and it's called a telomere. The telomere has been compared to the aglet of a shoelace. That's a word that I didn't know until I produced this show. The aglet is this thing over here. It's that protective cap at the end of the shoelace. The telomere, as I said before, it's non-genetic. It's repeats of this sequence, T, T, A, G, 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 over and over again, hundreds or even thousands of times. That means that that DNA can be lost because it's not genetic, it's not important. But what that means is that over time, your telomeres are shortening. Mine, as a 62-year-old man, are shorter than yours because you're probably a little bit younger than me. So here's how telomeres start. After a certain number of cell divisions, they're shorter, and they're shorter. Note that the amount of genetic DNA, the non-telomeric DNA, stays the same. It's only the telomere that's shortening. And at a certain point, when the telomere becomes too short, the cell becomes senescent. It stops dividing and eventually dies. That limit to cell division caused by telomere shortening, that's the Hayflick limit. Now, not every cell is subject to this limit. There are certain cells that are in our tissues that are called stem cells. Those cells have a unique ability to continue to replicate, and they become a reservoir of new cells that are used for regeneration and repair. Stem cells don't obey the Hayflick limit. How do they avoid this? They have an enzyme that's active. All of our cells have the gene for this enzyme, but only some of our cells are expressing those genes. That, in this case, are stem cells, and they're producing an enzyme that's called telomerase. And telomerase is an enzyme that is capable of extending the telomere. So in stem cells, telomerase is active, and it basically sets up the telomere to its appropriate length so that those cells can indefinitely divide. There are stem cells found in various tissues, and most importantly, the germ cells, the cells that give rise to sperm and egg cells, those are also cells in which telomerase is active. And that means that these cells reestablish the normal telomere number from generation to generation. Cancer cells also have the telomerase gene activated, and that makes cancer cells within the body not subject to the Hayflick limit. They're in some ways in your body essentially immortal, though they'll die when you die if you succumb to cancer. I know that I said that in a kind of dark way, but the main thing is that cancer cells can also be cultured. And when they're cultured in a culture dish, they're not subject to the Hayflick limit and they can live forever. There's a line of cancer cells that are called HeLa cells. They're named for Henrietta Lacks, who is an African-American woman who had ovarian cancer. In 1951, some of her cells were extracted and became the basis for a cell culture line that's widely used in research. This story of Henrietta Lacks, how her cells were taken from her without her consent, how none of the riches passed to her family for this enormously profitable cell line, is imbued with racism and health inequities. And Rebecca Skloot wrote a fantastic book about it that I can't recommend highly enough. It's perfect for AP biology students because it explains the biology, but also puts it in the social context. The Immortal Life of Henrietta Lacks, I can't recommend it strongly enough. Hayflick's other accomplishments include things like vaccine development, cell culture technology. He created an important line of cells, not the HeLa cells. He was responsible for advances in the use of microscopes. He was very important in developing intellectual property rights that apply to scientists, and he spent his life devoted to fighting all of the pseudoscience that's about life extension that you can see all over the internet, in newspapers. Hayflick knew that we are mortal, and we're mortal because of the limit that he identified. So now you know about the Hayflick limit, and here are some key takeaways. Most cells can only divide about 50 to 60 times. That's the Hayflick limit. The Hayflick limit is caused by the shortening of chromosomal caps that are called telomeres. After reaching the Hayflick limit, cells become senescent and die, and stem cells and cancer cells bypass the Hayflick limit through the activation of the telomerase gene, which rebuilds their telomeres. That's biology in the news for this week. If you have any questions, again, I'm sorry I can't be here live, but please leave them in the comments. I promise we'll answer them. Let's move into our song of the week. We've been talking about why cells stop dividing because of the Hayflick limit. Let's now look at how cells do divide with my song, Mitosis.
Guys, I want everyone's attention. We're going to go over what we did yesterday. We're going to go over the stages of mitosis. All right, everybody ready? All right, let's go. Interphase of cell division's longest part. Nuclear membranes intact as it starts. The cell's growing, cytoplasm flowing, chromosomes get duplicated, DNA gets replicated, chromosomes are spread out so they can't be seen distinctly, but note the nucleolus, the ribosome factory outside the nucleus, the two centrosomes. They later make a spindle which will pull apart the chromosomes. Prophase follows, the chromosomes condense, each is made of two sister chromatids like an X. Each sister is a clone, the closest of kin, and a centromere connects them like Siamese twins. The nucleolus disappears and melts away as the cell takes a ribosome production holiday. The centrosome separates, starts spindle formation for separating chromatids and cell elongation. Mitosis, chromosomal ride in a pro-meta and a telophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In late prophase, pro-metaphase, the nuclear membrane disintegrates. The centrosomes migrate to the cell's opposing sides, and between them the fibers of the spindle wend and wind. The spindle's made of microtubule fibers which attach to chromosomes. A kinetic causes a protein patch that serves like a handle that the fibers can grasp. When they pull apart the chromosomes, splitting them in half, the spindle moves the chromosomes with nudges so fine into linear formation on the 50-yard line, a location equatorial, defined in metaphase, where the chromosomes are lined up on that middle place. Mitosis, chromosomal ride in a pro-meta and a telophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. The spindle fibers pull on the kinetic cores. A cellular, molecular, mitotic tug of war. The centromere snaps, sisters get separated. Now these chromatids are chromosomes. They've been upgraded. The snap and separation defines anaphase. The A for apartness, for moving different ways. Kinetic core, spindle fiber separate the sisters. See, I'm waving goodbye, calling out I'm gonna miss you. And the other spindle fibers pushing grapple like felons, make the cell elliptical like a watermelon. In telophase, membranes form around the chromosomes that spread out as a nucleoli like come on home. Mitosis, chromosomal ride in a pro-meta and a telophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In animal cells, there's a ring of microfilaments that form at the equator and they cinch themselves in tighter till the cell is in two pieces. Yeah, in animals, that's cytokinesis, but it's different in plants. And then the cell divides by building a new cell wall from the inside as the Golgi sends vesicles with cellulose goo, which makes a plate. Then a wall divides a cell in two. And instead of one mother cell, we now have daughters too. Identical twins, kinda old but kinda new. From your single cell beginning, this is how you grew. And for single celled eukaryotes, it's reproductive too. Mitosis, chromosomal ride in a pro-meta and a telophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. Mitosis, chromosomal ride in a pro-meta and a telophase divide. Hey guys, eukaryotes go from one cell to two. Mitosis, alright, how cells renew.
And now it's time for the weekly quiz on learn-biology.com. Let's go there now. We're going to the Unit 1 review page as we did last week. We're going to go to multiple choice quiz 1, and we're going to make this a little bit bigger. And this week we are going to choose questions about carbohydrates and lipids. Notice there are 2 multiple choice quizzes. The other one has another 4. Which of the following includes molecules used for waterproofing, energy storage, and cell membranes? Choice 1, carbohydrates. Choice 2, lipids. Choice 3, proteins. Choice 4, nucleic acids. You got 5 seconds. 3, 2, 1. The answer is lipids. Nice job. Lipids include fats, oils, phospholipids, and waxes, and their functions include waterproofing, energy storage, and making up cell membranes. Question 2. The diagrams below represent various types of biological molecules or subunits of biological molecules. Molecules that would be categorized as lipids include: Diagram A shows 3 linked amino acids. Diagram B shows a molecule that consists of a nitrogenous base, a sugar, and a phosphate. Molecule C consists of 2 fatty acids connected to a glycerol and a phosphate-bearing head. Molecule D consists of 3 fatty acids linked to 1 glycerol. Which one or ones are molecules that would be categorized as lipids? Is it only A, only B, both A and B, A, B, and C, or C and D? I'm going to choose the wrong answers just to show you what happens on learn-biology.com. No, A is a polypeptide protein. We'll learn about that next week. B is a nucleotide, one of the monomers of nucleic acids, also an important molecule called ATP. But you're right about C, which is a phospholipid. We'll see this question again. Next question. Which of the molecules below is a key component of cell membranes? Number 1 shows a fatty acid. Number 2 shows 3 fatty acids that are bonded to a glycerol. And number 3 shows 2 fatty acids bonded to a glycerol with a hydrophilic head that has a phosphate group. So which of the molecules below is a key component of cell membranes? 1, 2, or 3? And the answer is 3. Excellent. 3 is a phospholipid, the key structural component of cell membranes, shown at A below. And let's go to the next question. 4 different biological molecules are represented in the diagram below. Molecule A is glucose. Molecules B, C, and D are composed of glucose subunits that are covalently linked together. If molecule C, 1, originates in a plant, and 2, consists of glucose molecules connected by bonds that allow that molecule to be broken down by humans for energy, then that molecule must be, is it cellulose? Is it glycogen? Is it albumin? Or is it starch? And the answer, molecule C, must be starch. Fantastic. Next question. Now we've done 4 questions. There were only 4, but in learn-biology.com, the quizzes lead to mastery. And here we see a repeat of a question that we saw before. The diagram below represents various types of biological molecules or subunits of biological molecules. A has 3 amino acids. B has a nitrogenous base, a sugar and a phosphate. C has 2 fatty acids, 1 glycerol and 1 phosphate-bearing head. D has 3 fatty acids and 1 glycerol. Which ones would be characterized as lipids? Only A, only B, both A and B, B and C, or C and D? And we learned last time that from the hint, it's got to be C and D. Excellent. Both C and D are lipids. C is a phospholipid and D is a triglyceride, the kind of molecule that makes up fats and oils. Let's view the summary report. In finishing this 4-question set, you entered 1 incorrect answer. All 4 questions were about topic 1.4, carbohydrates and lipids. Let's hear a word from our sponsor, learn-biology.com. Are you or your students struggling with AP Bio? With learn-biology.com, students get the skills and confidence they need to ace their biology class and the AP Bio exam. Learn-biology.com is 100% aligned with the College Board's AP Bio standards. It's approved by the College Board as a textbook alternative. Learn-Biology has interactive tutorials with readings, videos, flashcards, and quizzes that teach students everything they need to know about every topic in AP Biology. It has multiple choice questions, click and drag diagrams, identify the part exercises, free response questions. Students get positive feedback for correct responses. They get hints and repetition for incorrect responses. Student learning guides are present for every topic in the curriculum, and they tell students exactly what to do. Learn-Biology has everything you need to crush it on the AP Bio exam. Our library of interactive multiple choice questions, flashcards, and free response questions will teach you the material that you need to know so that you can review in a timely way and crush the AP Bio test. If you're a teacher, there's student performance monitoring so that you know what activities your students have completed. Free trials are available for teachers and students. Go to learn-biology.com and learn more. You won't regret it. So here's your plan for this week. Want you to go to learn-biology.com, then courses, then AP Bio, then go to Unit 1, Chemistry of Life. Last week we talked about the properties of water and some introductory biochemistry. This week we're getting much deeper, going into depth about carbohydrates and lipids. Spend an hour. You will be amazed by how much you learn, by the fluency that you develop related to biology, by your ability to talk about biology and really explain the wonderful concepts that you'll be learning about this year. If you're on a different curricular pathway, then learn-biology.com has the support that you need for any sequence. Learn-biology.com is the place to go for learning, and for more learning, what I want you to do is come back next week for another episode of the AP Bio Show. Please subscribe and watch this next video, which is going to teach you even more about the biology that you need to succeed. Thanks so much for joining me. I'll see you next week.